All right, I think I'm going to start. So I'm David Aragao from Diamond Light Source. I'll be pleasure to share this afternoon. It has been an incredibly good meeting, at least for me. I hope for you too, for all of you as well. No pressure on the speakers this afternoon to stay at the same level. No, I'm joking. We have a really good series of 4 speakers this afternoon. We will. The title of this session 7, the last one for this meeting, is More Ways to Characterize Your Sample. We'll be hearing about kinetic crystallography, time resolved. We're going to hear about placing fragments using anomalous signal, neutron diffraction, and also quantum refinement. So a little bit changing your gears a little bit over the afternoon, but I think really, really interesting. Thank you. Let's start this. So the first talk is by Nicolas Caramello about kinetic crystallography versus time resolved crystallography. If you don't mind to press the start, and the podium is yours.
Yep. Okay. Hello, everyone. So I'm Nicolas. I'm a recent postdoc at the IBS in Grenoble in the synchrotron group. And my project involves both cooperation between the University of Grenoble and the University of Taiwan, the National University of Taiwan. And in keeping with the title of the session, I'll be explaining how you can characterize your sample out of its resting state to study its dynamic experimentally with time-resolved crystallography and other methods. And maybe I can start by explaining why you would want to characterize your sample out of its equilibrium. We take the example of an enzyme here. In order to understand how it works, you must be able to observe very often the conformational change that go with the active site opening for the substrates to bind, the closing to catalyze the reaction, and opening again, for instance, to release the product. So you really have to solve the structure of the enzyme out of its resting state equilibrium and study its anharmonic movements. And historically, you can sort of separate 2 ways to tackle this problem. The first one has been, that I will refer to as trapping, has been finding a way to halt or slow down a reaction so that you can populate a reaction intermediate in a crystal. It started very early in crystallography, and it sort of peaked at the end of the 90s and beginning of the 2010s, from the end of the 90s to the beginning of the 2010s. And then second way to tackle this problem is to record crystallographic data as the reaction is going on in real time in the crystal. That's often referred to as time-resolved crystallography, and it's really undergone a revival since the beginning of the 2010s with the rise of crystallography at X-ray free electron lasers. So yeah, this is trapping, this is time-resolved crystallography. They all have strengths and drawbacks, and I'm going to detail those by explaining a few principles of these techniques. Starting with time-resolved crystallography, because I think it's what most of you are most familiar with, and it's been already presented during the study weekend. So most time-resolved experiments use the serial crystallography method, where you initiate a reaction in a crystal with a pulse of light, mixing with its substrate, whatever you want. And after a set delay of time, you record diffraction data. But in order to be able to transiently observe the system, you need the diffraction to be limited in time, because the duration of the exposure of your crystal determines very often the time resolution that you can achieve in your time-resolved experiments. Furthermore, when you initiate the reaction in a crystal, either you use a pulse of light and then only convert a part of your crystal, and you have a gradient of occupancy of activated state, or you mix it with something or keep the light on, and you will eventually have converted the entire crystal. It's become green, but in the meantime, you will have had an increasing gradient of occupancy. This means that time-resolved datasets often contain a mixture of different intermediate states. Here I have the gray state, which is the resting state, and the green state, which is the light activated state, as an example. There are 2 main X-ray sources where you can do time-resolved crystallography. The modern flavor of time-resolved crystallography was really born at X-ray free electron lasers, which have a particular design for their undulators, which groups the electron bunches and produces very tight femtosecond long and very bright X-ray pulses. That's very nice because it means you can achieve, in theory, femtosecond, hundreds of femtosecond as a time resolution. And it also means that your diffraction patterns are radiation damage free because the atoms in the crystal don't have time to move during the diffraction, which occurs in 100 or less of femtoseconds. You can also do time-resolved crystallography, time-resolved serial crystallography at synchrotrons, either by using the continuous beam of a synchrotron and moving the crystals in front of it and collecting diffraction patterns this way, or by creating pulses in the beam of the synchrotron by using a chopper. The best resolution that can be achieved right now with the upgrade of the SRF is microseconds, this way. But with this mode, you need a new crystal for every new measurement, and you need to find a way to fit them into the X-ray beam. You can categorize the crystal delivery systems that we use in time-resolved crystallography into 3 broad categories. Either you feed a slurry of crystals, viscous or liquid, in front of the X-ray beam and initiate the reaction in there and then shoot it with the X-ray beam, or you lay the crystal on a chip, move the chip in front of the beam and activate it ahead of the X-ray interaction point. And finally, there's a composite method that is often referred to as tape drive, where you lay your crystals on a moving surface that moves and goes towards the beam. All of these methods require microcrystals, so you need to find a way to grow these microcrystals, which can be challenging, especially if you want them size homogeneous. And even though they've gotten quite a lot better, they lead to very high sample consumption. So coming back to these strengths and weaknesses, high beam time and resource consumption for time-resolved crystallography. The interpretation of data because of the mixed states can be challenging. It's often, however, very close or as close as we can to physiological conditions, and we can access the intermediates in the order of the reaction because we're really observing the reaction in real time. So you can think of it as a golden standard, but a challenging experiment. And if we go to the first methods observed, you can think of trapping as finding a way to halt or slow down the reaction so that you can populate the intermediate in a crystal. One of the ways you can do that is to use an inhibitor or ration the concentration of substrate or cofactor. A good example of that is given in the study of an oxygen-dependent antibiotic catalyzing enzyme. Most of the authors are present in the room, I think. This enzyme uses dioxygen, and during the time-resolved experiments, the authors exposed the enzyme to dioxygen and noticed that upon binding, it caused the movement of a far away alpha helix, but they couldn't fully resolve the movement of this alpha helix because the turnover of the enzyme was blurring the state of the helix. So they resorted to using a surrogate for dioxygen, nitric oxide, which binds to the iron, but does not trigger the reaction and only triggers the conformational state. This allowed them to fully populate the outward conformation of the alpha helix and really resolve that intermediate. You can also achieve that by changing physicochemical parameters, such as pH, but most often the one that's been the most used is temperature. The simplest, that's because, as is nicely explained in this review, by lowering the temperature, you go through increasingly restricted conformational, increasingly restricting conformational change states. And you can think of it as lowering the potential energy landscape. Simple way to do it is to populate a steady state, for instance, and then freeze the crystal and trap the steady state, as was done a long time ago on this light sensing love domain, creating this carbon sulfur photoadduct. You might recognize this guy because I've used them in the introduction. A bit more elaborate, you can start the reaction and then freeze the crystal. So that one is closer to time resolved because you do have a delay of time. And there are now very efficient machines that have been developed to really do that, as for instance, the speed robot by the people of Hamburg. And then for light activated reactions, you can do something even more elaborate and start the reaction at different temperatures, effectively allowing the system only to go up to a certain point. This is what I call the freeze trigger approach. And a classical example of that is the bacteriorhodopsin. It's a proton pump from an archaea, giving the pretty pink color you can see in these lakes. It maintains a gradient of protons across the membrane, and the microorganism uses it to synthesize ATP. And the real biologically interesting question is, how does the light cause proton shuttling? So keep that in mind. This enzyme has been extensively studied by in-solution spectroscopy, and all of these spectroscopic intermediates, I, J, K, and so on, have been identified. And the timescale at which they occur has been identified too, but the structure of these intermediate was unknown. So using the freeze trigger approach, changing the temperature, but also the wavelength of the light they used to illuminate the crystal, they gradually found the structure of all of these intermediates. And in order to correlate the structure of the crystal to the known transient spectroscopy intermediates, they used in crystallo UV phase absorption spectroscopy, where you can match the spectrum of the crystal to the match of your known transient intermediate. That's an example of an isomorphous difference map, negative peaks in yellow, positive in blue, of a crystal that contains some of the M state. And one of the features, not very strong, but consistent over several structures of this state, is this positive peak here that the authors modeled as a transiently ordering a water molecule. This is quite important because that means that it can be involved in a transient proton pathway to take part in the shuttling. So coming back to our strengths and weaknesses, compared to time resolved, trapping is very resource efficient. When you're lucky, you need only one crystal. The crystal is often highly populated if you do things well. And potentially you can attain a higher resolution because you can use bigger crystal and solve the structure at cryogenic temperature, but you're operating far from physiological parameter and you don't know which intermediate state you've just created in the crystal and when it happens. However, as you can see with this checkerboard pattern, the two methods can be complementary. And here are a few examples of this synergetic use. Coming back to the bacteriorhodopsin, it's actually also been studied at X-ray free electron lasers in 2016. This is the study where they fed a slurry of crystal in front of, continuously, shot it with a green laser to start the reaction, and then shot it with the X-ray X-FEL beam to collect the diffraction pattern here, for instance, 16 nanoseconds after the beginning of the cycle. This is the result. So you can see these beautiful isomorphous difference map patterns building up over time within the entire protein. But the question is, which state or states were in those crystals at that time? And if you look closely at the isomorphous difference map, you might recognize this feature, the very seemingly weak feature that corresponds to actually what the trapping structure featured as a transiently ordering water molecule. And in fact, through the knowledge of the trapped structure, and because that feature was present in several intermediates, the authors of the study were able to identify it as indeed the transiently ordering water, and to conclude, they were able to conclude that these two time points, microsecond and millisecond, contained the M state of the bacteriorhodopsin. So the take-home message is that knowledge of previously trapped structures tremendously help the analysis of isomorphous difference map and the refinement of these partially occupied reaction intermediate states in time resolved experiments. But you don't even have to conduct these trapping experiments on purpose beforehand. Sometimes reanalyzing your synchrotron experiments can also be quite beneficial. This is an example of a study I took part in on the DNA photolyase. It's an enzyme that repairs DNA damage caused by UV. So these dimers of pyrimidine, for instance, this cyclobutane pyrimidine dimer in particular is the one repaired by the enzyme we studied. This is sort of its photocycle that has been studied also by in-solution spectroscopy. And this reaction is it uses a very redox sensitive flavin cofactor, meaning that it had to be studied in an X-ray free electron laser so that specific radiation damage would not parasite, so to speak, the data and cause changes that could be not linked to the reaction or just start the reaction altogether. And I'd like to pay specific attention to this last step here at the end of the repair of the DNA damage, where electrons are flowing back to the redox cofactor. It's called the back electron transfer. This is an overview of the enzyme. And if you look closely, between the DNA lesion and the flavin cofactor, there is a cluster of 5 water molecules that stabilizes the DNA. This came as a bit of a surprise because the previous structures solved at synchrotrons featured a 6 water cluster in green here, and it's consistent over several synchrotron structures of this protein. And in fact, the authors of these studies found out that the X-rays were reducing the cofactor during the reaction and initiating the DNA repair the same way that blue light would have had. And they found that out via also in crystallo-optical spectroscopy. And what's more surprising is that one of the time points isolated at an X-FEL, precisely 3.35 nanoseconds after the pulse of light initiating the DNA repair reaction, also features this 6 water cluster in red here. And when we studied this structure, when my collaborators studied these structures, they found out that this very likely corresponded to the back electron transfer step, which is happening stochastically, meaning that the redox cofactor here is in a mixed state. Some of it has been reduced again, and some of it has not been reduced yet. And the common point between the synchrotron structures and these time points is this mixed state of the flavin cofactor. So here, so that's why this one also features a 6 water cluster, despite this one being room temperature and these being cryogenic structures. So here is an example of really involuntary trapping. The synchrotron structure had captured a feature present during the reaction, and that can locally be considered as an intermediate. And having access to a structure that had this cluster of water at a higher resolution and more stable was really useful to interpret the different density patterns of the time-resolved experiments. So as a take-home message to my talk, there's more to kinetic crystallography, to characterizing the dynamics of your sample than just time-resolved experiments at room temperature. The very simple, so to speak, trapping procedures that have been used for a long time can really complement the time-resolved experiments. And they can, if you're lucky, access some of the reaction intermediates. But in order to make sure that those are physiological reaction intermediates and to place them in context of the reaction, you will need complementary methods, complementary biophysical methods such as spectroscopy and genuine time-resolved experiments. So I'd like to thank a few people, Jos Kemps for helping me understand the IPNS study, as well as Antoine and Sylvain from the IBS, the entire lab of Junpei from the University of Osaka, the lab of Manuel Maestrerena, in particular Yu Hei from the National Taiwan University, and the lab of Mingdao Tsai from the Academia Sinica, also in Taiwan, and the lab of Lars Oliver Essen in Marburg, in the University of Marburg. All of these people are part of our collaboration on the DNA photolayers. And I'd like to thank you for your attention.
Thank you, Nicolas. Really clear talk, elucidating. Do we have any burning questions? Start by making one myself. In this comparison that you were saying between an X-FEL time point and the synchrotron data, the synchrotron data was rotation, cryo-cooled crystallography.
Yep.
So essentially we were comparing serial with non-serial.
Yep.
Just trying to defend a bit the synchrotron because you can do serial in the synchrotron too, right?
Absolutely. You could also make the same arguments with a serial time-resolved experiment at a synchrotron and the cryo-cooled rotation data that you've collected also at the synchrotron. It's really a matter of preparing your experiment by trying to isolate these intermediates so that you know what you're looking for when you get the isomorphous difference patterns.
Thank you. We still have a few minutes for some questions. Oh, yeah.
Thank you very much for your talk. So you were showing the initiation of the reactions with light and the different strategies that you can use to trap your intermediates. So can all of these also be translated to like diffusion of a substrate, or is that more complicated?
No, not all of them. The freeze trigger approach, the one used for the bacterial rhodopsin, would be impossible to do with a substrate because you cannot diffuse it if the crystal is frozen. So that one, unfortunately, only really works for processes initiated by light. If you're very lucky, you might have a photo-caged compound that is really close to your active site and that you could activate uncleave at cryogenic temperature. But this is a lot of ifs.
Any more questions? All right, let's thank Nicolas again, please.